Hi everyone. In this video, we will explore CellTypist and another reference-based, single cell data annotation method. In the last video, we saw single R, which was based on the correlation, and then this tool is mainly, machine learning based approach. Okay. So yeah, again, so just to give a quick recap, what is reference based? You have a query you want to annotate, and you have, let's say, some reference data set. You try to map your query against that. It's like kind of alignment and transfer that label from that reference data set to our query. So but now this cell typist is a machine learning method. Means like that means it has trained various data set, the labels, and it trained a machine learning model, and that model is now able to will be able to classify your query data set. So let me explain little detail. uh, the classification ML algorithm is mainly logistic regression classifier. And furthermore, it is optimized by the stochastic gradient descent algorithm. That means, more, it will be the minimization of the error, So that way it tries to classify your query cell type into, it will give some annotation. Okay. And again, the only difference, like, it is Python based, single R was R based, and this is the main website. You can go over here. So this, if you go to the CellTypist, right, celltypist.org, this is the main homepage. So you can try on website also, and at the same time you can also do the Python based coding also. So we will see both. The interesting thing is like the models. Okay. So what is, like, they have the machine learning models. So just let me zoom out. So all the models are built on logistic regression framework. Okay. So what it means, like, they have the different models, like these are the models. For example, some CHCPC, this is human circulating hematopoietic stem and progenitor cell. Okay. So if you want to, so this is the dataset they have taken from this paper, and that then they have this data set, this data set, this data set, like that. You can see so many data sets are there. So depending on your data, you can choose a particular model or particular reference data set. So this, they have provided the model in the form of a pkl file, so which is nothing but the data that contains the all the what is the weightage and all. Okay. So just to explain you that in a text wise, so basically how CellTypist work is like, so they first do the training. Okay. Because the machine learning model, right? So they have taken the massive reference data set. So let's say, for example, they took some paper and they have some, let's say, 20 tissue and 18 studies, like that. So that's the whole data set, and for each of these data set they trained a logistic regression classifier. And so the reference now contains a compressed file that contains weight or coefficient for the 1000 of genes. So now when we run our data set, so it will take our data set and it will use those model and coefficient to classify what cell type it is. Okay. So that's how all the machine learning classifier works, right? So it compares and it tries to weight and bias it does, and tries to give you the correct level classify. Okay. So that's how it works. And yeah, so these are the different levels that are available. Okay. Alternately, what we can go, uh, so they are very nice, this thing, so you can annotate your own data set. So you can give it your email and you can choose a model, and what criteria you want to compare, voting yes or no. And here you can provide a CSV file or directly H5AD file, you can give it here. Okay. And basically they also need the data, I think, in the form of a log normalized, I think. Yep. So we need a log normalized expression matrix as an input. Okay. And at least up to 1000 counts per cell they need. Okay. So that's when normalization should be done, so that the total cell count should be the UMA count should be normalized to up to 10,000. So that's how we need to provide that data set. Okay. And so this, they have given some example. Let's say if you have some raw count A data, right, the Python annotation data format, so you can simply do this one. So you can say that how much target normalized value you want, that is like 10,000. So that means all our UMA count should be normalized against that value, and then you can just do a log transform. This is simple log normalization. You can do it. Okay. And detailed model information can be found here. I think this will land to that same page only. Okay, let it be loading, and this is the GitHub repository. Let it load. So they have, you can try the this notebook, you can open in Google Colab and try to do it. Alternately, you can install pip install CellTypist, and then you can run the command. Okay, so I'll just show you with one example how you can use CellTypist for cell type classification. How does it work, and that will give you some idea. And same, if you're very not comfortable, you can come back here and try to do submit the job here, in the home page you can try. Okay, so this is what the Google Colab. I am opening this one, the first one, CellTypist. Okay, we'll follow the same one. So first we need to install pip install. I have already installed here. It will take some time. So yeah, so it is installed. We need to install the CellTypist package, pip install. Then we load this library, Scanpy library, and then we can from first you import CellTypist, and from this first let's import the models. Okay, so this is our query dataset. We want to download it, and this is downloaded from this H580 format, some the URL. And so yeah, so basically this data set includes how many cells? 2000 cells and a data of around 18,950 genes. So that's why if you do shape, you will see 2000 by a 18950. Okay, that many cells. But now the interesting thing, like, this data is log normalized. Okay, that means every cell's data is normalized. So that's why if I take the row of sum, we will get 10,000 value. That is what it is expected. Okay, so if I, this is what, if you do right, the first cell, the total sum of expression is 10,000, second one is 10,000, third one is 10,000, everything is 10,000. That's why all cell are now normalized, so that the total sum of the expression value is now 10,000. So that way, so the expression matrix is pre-processed and required. That's very important, that edge log normalized expression to 10,000 counts per cell. Okay, this matrix can be alternately stashed in .row.x. That's okay. So this is how you need to prepare it. So ideally what I would suggest, if you are not comfortable in Python, if you are in R, so you can take R data and you normalize in R and make sure that you log transformed value you get, and then you can load and Scanpy the same one, and no need to transform it, and directly you can do sc.read, and you can store in that format. H580 you can load in that format. Okay, so just for they have also, for this query dataset, they already they have given some type which will help us to compare if you are using this model how much accurate it is. So that's why some already they know the for cell 1, let's say it's a plasma cell. For some cell 2000, this is progenitor set. This is not a PVMC dataset, this is a dataset from some other dataset. That's what you need to, it's a very different dataset. Okay, so now what kind of models are there? So this download models means like we are using the models, and this is the, once you download it, it will download in some path in your this Google Colab workspace, or if you are doing it locally, it will be in your local path, and you can see the description. This will again the same what we saw here, right? If you go to, what is that, models. So these are the model name you will see, and what it is, the same information you will get from here. Like for this pkl file contains what. Okay, so now there are 61 rows are there, means that means 61 such models are available. Okay, or data sets are available. So now let's say we want to compare our dataset against this immune all low, for example. Okay, so you can do models dot model dot load. You can load this one. So now this is the model, and just you can print it. It will tell like this model contains 98 cell types, and this has around 6,639 genes. Okay, so that means the dimension is, if I cell by gene, if you do 98 cell types, not number of cells, types, and 6,639 are the genes are there, and it is combined from 20 tissue and 18 studies, like that. Okay, so that's why if I do the cell types, so these are the different cell types what we get, 98 cell types, right? The data, these are the cell types. And now this is what the important part, this is the one line command. That's where you are transferring the annotate, here giving the query, and here choosing the model, and here currently we are giving of true or false. So now yeah, so by default majority voting is false, because we don't want voting true, but it will take some time. So for now, just to I will just saying that I don't want voting, so that it will run quickly. Yeah, it ran it. So we got this all these predictions. So now if you just do the prediction dot model predicted level, so it now got predicted level. Okay, so the original one what we saw in the original we saw, right, that is cell type was the original one. So now we can do the comparison as well. Okay, and yeah, so this is what we can further convert into annotation data format. So prediction.to a data. So this is what a data format we got. And if you are doing this one a data dot observation, so you will see like, this is my cell one, this was my original metadata was there, and this is what the predicted level what I got, and the confidence score what according to the, this is the logistic regression, the odd score what we get here is the value is more confident we are. Okay, so in addition to this metadata information, it also gives some graph constructed from the over clustering. Okay, so that graph is nothing but the UMAP coordinates also it provides, which we can use to do this UMAP. Okay, so so because I've not done that neighbor, right, so I'll have to run this neighbors first. Okay, there was some error, I just fixed it. So basically, yeah, that's what we need to compute neighbors, right? So this is how you will do it. Neighborhood, this function we call, and it will do the neighborhood calculation. Okay, it will calculate again. Okay, then we can do the UMAP projections. And now we can do the visualization. So basically here we are drawing the same color with respect to the cell type, and also we want to do the predicted level. So you can see that the original is this one. So for this one macrophages, here we got intermediate macrophages and other things. So it is more granular annotation you see here, but yeah, it's very difficult to see. But that's what it is saying, we need to compare. So that's why instead of this plotting you can rely on this kind of thing, and then you can manually do the plotting as well. Okay, yeah, and this gives like confidence score as well, like dot plot kind of thing you can do, and the reference is, let's say, cell type the original one and against the predicted level if you do. So this will show the confidence, like mean probability is the red one, and how many percentage of cells they are expressed. Okay, so this gives another information mainly of dot plot, like macrophage is the reference and macrophage is the uh, query. So, and the value what we get is the probability size and like that. Okay, so here is the round means color is, sorry, red means more with the probability score, and blue means these are less probability. So that's where all the colors what you see are mainly, the red one means the probability of that means the confidence is very high. Okay, this is how you can do it. Alternately, you can also choose a custom model. So for example, let's say you do not have a model, then how you can do it. So this is what they have given example. This is the query data set, and so you can use the train function, and you can use what cell type in that query data, and you can do that. So that's why you will get a new model, and now against that model you can save it in a pkl format, and you take that model and you repeat the process. Okay, so that way it is also useful if you have some data in that matrix format, like cell by uh, cell by sorry, gene by cell, or cell by gene in Python. They are just confused with R. The metrics if you have, but that should be log normalized. Then you can you call this the logistic regression training method to do that. Okay, so in short, like it's not that complex if you're comfortable with Python, and that is mainly so what it does, like or alternately best is like you can choose and upload a simple CSV file, and or H5AD file. CSV means not just marker name, because it should be the matrix format, right? The gene by sample, the log run with the expression value. I think ideally I would suggest like you can generate from R and you just upload it. If it works very small size, then it should work. Okay, so yep, I think that's all on this one. Okay, so yeah, we saw CellTypist. So far we covered the marker based, and we moved to reference based, and we covered single R, and I have talked the CellTypist. As I said, assume some issues I'm getting, I'm not getting time to resolve that. So whenever I complete it, I will upload that azimuth, or you yourself can try it out. Okay, so again, that's mainly projection based method. But now These two are the older method, but now we have because of the AI evolution, so we get some AI assisted annotation methods are there. So there are some popular tools are there like CellLlama, MLLM, CellType, Cassia, and more tools are coming. That's what, and I will give you some glimpse about like how to use this LLM or this new AI based method as well. So that's what we will see in our next video. I think we will start with the CellLlama, then probably MCellType, and then we will move to the Cassia one. Okay. I hope this CellTypist gave you some idea about like sometimes if you have the option to try this ML based method, you can try it out because they are very trained against the reference data set. Okay, easy, and it is very fast as well. Uh, uh, because single R is morely correlation based, right? So correlation there is not much statistics, but here ML based method, you may get some more good accuracy. So it's worth trying. That's what I thought to include that approach as well. I hope this video was useful, and if yes, please give me a feedback like how did you like it, or any mistake I did, please excuse me. Okay, so anyways, I will see you in the next video. Thanks for watching.